Now we will start our webinar. I request Dr. Umarani Madam, Associate Professor of Chemistry, Government Arts College, Coimbatore, to welcome the gathering.
Hello participants, a very good morning. I am Dr. Umarani, Government Arts College, Coimbatore. Hope you are all fine and fit. We know that COVID cases are worsening day by day. It is actually tightening its grip. We are advised to be at home. Because of this pandemic, the entire educational system is helpless and wanting for the pandemic to slow down and end. In this difficult time, we got adopted to a new normal life. So how we are going to lead this new normal life is a big challenge, especially students. They are very anxious, much bothered about their future, and are really confused. So to maintain the flow of academics, we should take advantage of this opportunity. Webinar is a good initiative. In this concern, our department faculties are conducting various programs. Today's webinar is arranged by our lecturer, Dr. PONNUSWAMY sir. More than 700 have registered. So it's a very good response, and we are very thankful for the trust you all keep on us. On behalf of the PG and Research Department of Chemistry, Government Arts Coimbatore, I welcome our Chief Guest, Dr.U.P.SENTHIL KUMAR, Senior Vice President, R&D Center, Orchid Pharma Limited. Welcome you, sir. Sir, I welcome our principal, and we are very thankful for her continuous support. I take privilege in welcoming all the faculty members from other universities and colleges, and our HOD, Dr. PONNUSWAMY, who works for the upliftment of our department. I extend a warm welcome to my colleagues, other department faculty from our college, research scholars, students, and all participants from all over the world. Finally, I wish to extend a warm welcome to Dr. Mohan Raj, our alumni, for his technical support, and Dr. Ratna Kumar, Assistant Professor, BBA Department, Manoj University Constant College, for his support in YouTube link. And I think surely the seminar will be meaningful and useful to all the participants. We are actually going to have a nice time. We'll be running a live question and answer session at the end. You can also type your queries in the chat box. And now our HOD, Dr. PONNUSWAMY, will introduce our chief guest to you. And again, a warm welcome for those of you joining now. Health is our wealth. Stay home, stay safe. Thank you. Over to Dr. PONNUSWAMY.
Very good morning to everyone present in this meeting. I thank Umarani Madam for the very nice welcome to our participants. And really, I deem it a great pleasure to introduce Dr. U.P.SENTHIL Kumar to our audience. Dr. U.P.SENTHIL Kumar completed his master's degree in chemistry at American College, Madurai, and he pursued doctoral research at Bharatdasan University under the guidance of Dr. Ramasubbu Jayaraman, focusing on synthesis and conformational analysis in relation to the anti-cancer properties of N-nitroso heterocycles. And for his PhD, he has passed both CSER net as well as GATE. But importantly, he is All India topper with a score of 99.99 percentile score. But he did his PhD using the Dr. Krishnan Fellowship. It's one of the very prestigious fellowship offered to a few in India. And he has used that fellowship for his PhD. And we fondly call him as UPS. And in my academic career, I followed him for more than 12 years, from +2 at Diamond Jubilee Higher Secondary School from Gobichettipalayam, and BSc from Gobi Arts and Science College, and MSc from American College, and PhD under the guidance of Professor Jayaraman at Bharatdasan University. More than 12 years I followed him, and he is really a very intelligent person, and a very humble and intelligent person. And let me say a few words about his achievement as a scientist. He has joined after his PhD. He has joined as a scientist at Torrent Research Center, Ahmedabad, where he was involved in process research and drug discovery research on ACE inhibitors. In 1997, he joined Orchid Pharma Limited and worked on developing manufacturing technology for active pharmaceutical ingredients of beta-lactam antibiotics or non-antibiotics and antibiotic drug discovery activities. He has developed several generic products for regulated markets, successfully overcoming regulatory oppositions, challenges, and patent litigations to launch ahead of patent term expiry. He had designed patent strategy to develop and launch products in regulated markets and successfully appeared for patent depositions at the US court for Para 4 FDA products. He secured about 100 articles patents. You think about that. It's not easy to get around 100 patents, which include invention on new drugs, products, processes, rearrangement, novel polymorphs. So that's the reason Orchid secured National Intellectual Property Award. That is the top Indian company in patent to get the award by Indian Intellectual Property Office, Department of Industrial Policy and Promotion, Ministry of Commerce and Industry. A man behind this achievement is Dr. U.P.S. And he has also, in particular about his achievement in this field, he developed a novel beta-lactamase inhibitor and out-licensed to Electra Antibiotics Germany. Successful clinical trials demonstrated superiority to the widely prescribed piperacillin and tazobactam indicated for the treatment of complicated urinary tract infections and also pylonephritides. It is the first such drug candidate in this antibiotic category to be out-licensed in the country. It's a great achievement for him. His responsibility includes leading intellectual property management activities at the R&D center, catering to the need of research units, process research, and developments, manufacturing and business development groups at Orchid. So, more than this, actually, he is more than a great scientist, he is a great human being, very humble personality, very simple person and down to earth. And he has offered many foreign abroad chances consistently, but he denied it, and he has been doing a lot of community service to our nation. And he has been involved in Dr. APJ Abdul Kalam Foundation as a key member, and he is doing a lot of service through this. And of course, you all know that we are running a foundation under the banner of our Professor Ramasubbu Jayaraman, and it's the 9th year we are doing the NET research drive program actually, and more than 200 students have cleared the NET using through this program, and many of us involved in the project though, but the project was written by Dr. U.P.S. actually, and with this, and he is also a classmate of our own colleague Dr. Vijayalakshmi at Gobi Arts and Science College. And with this small introduction about him, I request to I hand over the session to Dr. U.P.S. and we fondly call him U.P.S. uninterrupted power supply. And one thing I wanted to tell you, he along with our seniors are responsible for creating a conducive atmosphere for research at Bharathiar University, though we have very, very minimum facilities. Thank you, Senthil. And now I hand over the session to Dr. U.P.S. Please, I request the participants to mute your audio. Mohan?
I think I can go to present. Okay, sure. Anushka. Did it come now? Anushka. Okay, one second. Let me do this. Has it come now? Fine. Shall I start? I think voice and sound is clear. No? Wonderful, wonderful. Let me start. Very good morning to all of you. And I should thank Pons, with whom I am associated from school days, from +2, and we did chemistry undergraduation in Gopi Arts College, and then American College we did for PG, and then PhD we did at Bharati Dasan University under Professor Jayaraman. The entire period was excellent, and we cherished the relationship, and that was an exciting period of learning. We never felt that we struggled to learn. As Pons said, even Pons also scored very high score. And we never felt that we struggled hard to get the score. We enjoyed, at the same time we learned. And I'm really thankful for Dr. S. PONNUSWAMY, Associate Professor and Head of the Department of Chemistry, Government Arts College, for arranging a series of webinars in which he invited me also to be a resource faculty in one of the talks, and today I'm with you. And the important thing is that we are going to discuss something about the drug discovery and microorganisms and challenges and opportunities to combat COVID-19. COVID-19 is not the only issue. There are going to be a lot of surprises. And this 21st century is for the scientists, especially for chemists and biologists and pharmacologists. And WHO, a couple of decades back, announced that this 21st century there will be the challenges for the science, and also they listed top 10 priorities for this 21st century, and one of in the top listed items is infection, pandemics and epidemics out of microorganisms, and that talks about the importance. And today, everybody in the world, every country in the world, whether it is a developed nation or underdeveloped or developing country, every country in the world is looking at the scientists. When are we going to have the drug? Either today or tomorrow, how fast we can get the drug? And never in the history, society has been the drug. The first society has not seen the pandemics. They have seen several such pandemics has happened and killed and destroyed even civilizations. Entire civilization was washed out because of certain pandemics, and several crores of people were washed away with the pandemics in early centuries. Even in the last century, 1918, we had influenza. Even at that time, the society did not look at the scientists when we'll have the drug, because allopathy did not become famous, because it is just 200 years old, allopathic medicine. And because of the invention of the penicillin in 1942, in 1930s we had sulfur drugs, first set of antibiotics, and 42 penicillin was introduced by Dr. Alexander Fleming and other couple of scientists, and who are all Nobel laureates. Then people looked at allopathic as one of the potential drug discovery, and from there onwards, this has started going exponentially. Drug discovery through synthetic methods. Of course, it takes natural products as a inspiration. And then what happens? The treatments which were there for thousands and thousands of years, Siddha, Ayurvedic, Unani, all these things were called as alternate medicines, and primary medicine was considered to be, has been considered to be allopathic treatment, and this kind of new drugs from the labs by scientists and biologists. The problem is that it is not only just an economy of a country. Every country is struggling today. Country can fight. The America and China, India, they are ready to fight with other countries. They have prepared well in terms of for war. All technology, science is used. They are ready to. They face courageously to attack the other country and to defend their country. But there is a still bigger war that is going on with microorganisms. Even biggest countries, US, China, everybody is shaken because the enemy is within you, and you are only multiplying him, and you don't know how to handle him. And there's big distress, and a lot of stress is going on everywhere in the world. Countries are not able to manage it, and these smaller viruses can make us to sit at home for months and months. We don't know when it's going to complete. And second, this lead not only in a bigger level of economy, even if you go to the individual levels, people are starving for food. People are committing suicide because they don't have food. They don't have a job. This virus, which is not even visible to naked eye, which is not even having a life, which cannot multiply on its own, it takes as a host system. When it comes to enemy, it stays within the enemy. We are the enemy for it. They go into us and they multiply. They use our cells. They go to the enemy's place and they multiply and kill the enemy. And such circumstances, there is a big need we need to come up with a drug very fast. Who can do it? Chemists can do, in association with biologists and pharmacologists. But how fast it can be done, that's what we are going to discuss now. And when we talk about drug, we should never forget, even the world never forgets, that India is playing a major role in terms of producing and supplying generic drugs. Generic means those drugs for which patent expired. So until there is a patent which is there for 20 years, the company which innovates only can manufacture and sell. Whereas after that, anybody in the world can make. When many people make, competition comes and price goes down. In this scenario of high competition, India is producing the 20% to 25% of the entire world requirement of generic drugs. You can think about how good our chemists are, how good our biologists are, and pharmacologists and clinicians, who are able to make a drug, generic drug, which is equivalent to what is introduced by the innovator companies and is accepted by every country in the world. And the biggest pharmaceutical country is United States, which is holding almost 50% of the entire world share. And for US, we are one of the major suppliers. US procures generic drugs across the world. And from a single country, US is importing 40% of the drugs, generic drugs, and that country is India. Now we can understand the quality, standard, consistency, GMP, good manufacturing practice that is followed in India compared to any other country. Rest 60% drugs are organized all over the world, and 40% goes from India. And we can keep on telling like that. Even vaccines, about 60 to 70% of the entire global requirement is made by India and supplied. This is about manufacturing the drug and supplying to the world. That is also important. No? You may have a drug, but who will manufacture? What is the technology? How do you assess the quality? How do you assess the bioequivalence? A lot of things are there. In those areas, it's finite, but drug discovery is a separate arena, making a new drug. But before going to that, we should think about what is about this microorganism. The world comprises four major kingdoms. You have microorganisms. Next level, you have
Senthil, wait a minute. Zakir Hussain, please, you come out of that. You don't touch the presenting screen. Please come out.
Okay. Sure, okay. Yeah, I'll go back and present now. Yeah. So, I think it's coming now, correct? Yeah. So, is it audible and visible? Great. So, when you see there are four kingdoms in the world, we have a microorganisms which we are not even able to see. These are bacteria, these are all virus, and we have fungus and then protozoa. These kind of things are one set of organisms which are not visible to the naked eye. Next level, you have plants, and you have animal, and the fourth is one second. And fourth, you have human beings. We are thinking that we are ruling the world. We are ruling animals, we are ruling the plant, and we are also thinking that we are ruling the microorganisms. But truth is different. Actually, the world is ruled by microorganisms, which is once in a while, microbes shows that they are ruling the world. And we are running away. They are able to make us sit at home. They are able to close all the shops and even offices. And they take a more role than anybody else. No, so we can stop China from coming inside, but we could not stop these microbes coming into the country, not even into the country, but into us. You can see this. 100 years are over. In 1918, these beds, which were And this photo was taken in 1918 in a campus when the influenza attacked the world, where between 5 crores to 10 crores of people died. This is one such photograph. Even today, we see the same photograph. 100 years are over. We think that we transformed the world entirely with thousands of drugs and several hundred antibiotics of different categories. We thought that we are able to handle these organisms. For example, in earlier days, even a small cut in your skin, even a small disturbance in the skin could be leading you to death. The organism which enters inside can multiply and lead to kind of different poisoning of the blood. Finally, we died. But today we have a lot of treatments. We don't even go to hospital. We take an antibiotic at state. That is the level of growth we have got it. But viruses, again, this virus and bacteria again and again showing that we have not gone anywhere, because virus and bacteria, they are also doing drug discovery. They could be as small as anything else. Virus is hundreds of times smaller than our cell, human cell. But that virus is also doing discovery. It keeps on changing again and again and again. And every time we are shocked and we are running around to discover a new drug. That's the reason even after hundreds of years, these beds are even seen today. We are nowhere. These organisms are tiny, invisible, and they triggered, this notorious organism triggered several events that drastically altered the human history. One second. And they created mass deaths and destruction. Even at one point, 1/3 of the population, close to 1/3 to half of the population of Europe was erased out, and some civilizations were completely erased by the organisms. And it happens every century. Few incidents are happening. Not only that, we should not be afraid that it's always doing wrong things. And it is organisms who inspired modern medical advances. There are a lot of examples, and they are responsible for the industrialization. I can explain when you go forward. They are responsible for the world to become industrialized. So they played major role. In fact, not only that, they created the world. They brought the life into the world. Cyanobacteria are the earliest things which lived in the world. They were anaerobic bacteria. So entire earth, there was no oxygen. It was all anaerobic, and the cyanobacteria are single-celled organisms which lived in anaerobic conditions in the absence of oxygen. But they evolved, and when they evolved with the photosynthetic machinery about 2.5 billion years ago, oxygen was produced in the system, and evolution started. Several evolution steps started, and we started getting all the organisms, plants, animals, human beings, like multicellular eukaryotic organisms started. In a way, these bacteria are created the world. It's their world, and we are part of them. We need not think that, and we should not think that. No, we are ruling it. Rather, and then everything, smallest of everything, is actually created every living being in the world. And once in a while, they do some kind of testing like this. And in history, I will quote, there are about 7 or 8 major pandemic last 1,000 years, which really shaped the world, which changed the world, the way it was looking at, and changed even civilizations, which brought industrialization. In 1845, just about 150 years back, in Ireland, there was a fungus called Phytophthora infestans, and that led to Irish potato famine. That means, damage of all food materials by this fungus, so that people were starving to death. It attacked about 3/4, almost 75% of all the crops in Ireland was destroyed because of this fungus, Phytophthora infestans. Ireland was fast growing at the time. They were not even a crore. There are about 60 or 70 lakhs of people were there. It led to at least 10 lakhs of deaths due to starvation, because food was completely destroyed by this fungus. And another 10 to 15 lakh people fled away from the country to have some food and avoid the deaths. And this is an influenza pandemic. We are thinking that virus is responsible. but not virus alone. It killed about 5 to 10 crores. And it's not, that's the first pandemic. It happened in 412 BCE and 1357 and 1580 Asia, and it spread to Africa, Europe, and several countries. Even though there are several tragic pandemics happened in last couple of thousand years, this 1980-90 was the highest, with an unprecedented death toll of between 5 to 10 crores. All of us were thinking influenza virus killed everybody, but it is not really true. What the communities, scientific and medical communities, are believing that it is due to bacterial pneumonia. What happens? Virus, the influenza virus, enters into your lung and bronchial tissues and destroys. Once these are all destroyed, this normal nose bacteria and this kind of throat flora, there is a very simple bacteria which otherwise are harmless. They invade into the lung tissue and cause the disease and expand and spread and causing the death. Virus entered and it paved a way for bacteria, and bacteria entered and destroyed it. In a way, they collaborated, they joined together, and there are caused mostly this kind of pneumonia caused by Streptococcus pneumonia and here Haemophilus influenza, Staphylococcus aureus, and Streptococcus species. And Streptococcus pneumonia is one of the most notorious bacteria. And you know, this 1942, 1928, it was first identified, and 1942 it was introduced. This penicillins by Alexander Fleming, Howard Florey, and Einstein. It's a groundbreaking human drug, and because of this invention, many things happened. Society has changed. Allopathy has grown. People started looking after new drugs from allopathy. Until then, it was not. Maybe one of you can mute that. Yeah. So allopathy has got a new shape because of the penicillin.
Sorry, madam, please mute your audio.
So allopathy has got a new shape, and the world has got a new development. Mass deaths have stopped, and the science was given a focus in every country. You know one thing, in 1918-19 we have seen that this influenza pandemic killed 5 to 10 crores, but just before that, 1914 to 18, there was a world war which caused a death about 2 crores. You know, in the World War I, out of 2 crores people who have died in the war, they didn't die because of the war. No? They got small cuts and scratches in the skin, and that developed into a pneumonia, into a bacterial disease, and finally they died. Most of the people died not at the war, but because of the infections that happened. Now, and that killed them. So after this discovery of penicillin in 1942, you can see that in the World War II, there are about 6 crores deaths, but death due to the infection was less than 2%. So that is a major change that is brought by this penicillins. Had penicillins was not invented at the time, then death could be more than 40 to 50 crores during the Second World War. And that has made a major change. It has brought in this discovery. And another worst case is this bubonic plague, means black death, caused because of the bacteria called Yersinia pestis, which is is actually spreads by fleas. Fleas from rats and mice and squirrels, infected animals. If they sit on us and they take the blood and they spread it, and it was the worst in the history. In the first pandemic happened in 541, and it was there for 200 years. So we cannot think that this COVID can stop in short period. The thing is that it will not die. Now virus will lie somewhere, because it is not that it will die something. Somebody should live, then only they can die. It's a particle. It's a protein particle, which means it can lie somewhere and again it come out, unless it is eaten by something on the surface. And here it killed even that 541, about 1,500, 600 years back, it killed 202.5 crores of people. That period, that could be a major population. About 1,500, 2.5 crores is a big thing. And the second and the most infamous pandemic is this Black Death. It started in China. In China it started in 1334 and spread to Europe. So those periods there was no flight. Only ship was there, or roadways. At that time, the ship started from China, And then they reached Europe. When they reached Europe, the people, the sailors who were infected in China. When the ship reached this Sicily, that is a part of Italy, it's a small island in Italy. When the people entered in Italy, they found that all sailors were either dead or about to die. So from there, people unloaded and then spread to France and then to North Africa and Italy. That killed about 60% of population of Europe. Not only human beings, livestock like goats, pigs, chicken, cows, and sheep, everything was affected, and that was not under count. Only human count was 60% of population was eroded by this pandemic, Black Death. That's what is called in history. It's not only the negative thing, and because of that huge shortage of manpower created, and that led to major economic and technical modernization. So all industrialization started, and it assisted. This Black Death assisted emergence of a Renaissance period, and because of the industrialization, the entire world also got a benefit. Today, whatever we are, even our growth is our technology, our industrialization, all again driven by these microorganisms. And the third happened in 1860, and about 1 crores death happened. But the problem is, it was there for 20 years. For example, if this corona lasts for a long time, and we have to live with mask, and that will become our style itself, like shirt and pant and shoes, and we'll also start wearing masks. It serves for over a period. We may find a drug for this corona, but corona too will come. This corona, it is not appearing this year. It is dated back to 1957. Corona is not a new virus. And many coronavirus are as simple as your simple cold, and this is the one which is having a different effect, and that's the reason we are all in trouble. Otherwise, there are various versions of corona which are harmless, like adenovirus. They are all harmless. And here the carriers were rodent-borne fleas. So in this plague, at least you should be happy that in your case you can control it. Unlike this kind of, like mosquito spread something, all the fleas are spreading this bacteria. At least in this particular corona, one of the important thing is that unless you allow it, it will not go inside. Whereas in these cases, like bubonic plague or influenza or many other cases, you don't have a control. No, how can you control that? No mosquito should bite you. You have to go outside the society, somewhere something will come, or they will come and say. In this case, there are only 3 channels with which it can go inside: mouth, nose, and eyes. Your entire body may have a contamination of virus, but still you can prevent from entering into you, unless otherwise, you use your hands to touch any of these things. Sometimes you get irritation, immediately your hand goes and touches eye or or mouth. So whenever you need it, please see that you have to touch, you have an irritation, you have to eat. Go and wash with the soap and then come back. Nothing will happen to you. So, on good side of is that you can control it, as well as you do not use your fingers to touch any of these 3 entry into the body. So skin is a wonderful protection. So through skin, it cannot go inside. Whereas you also should cooperate with virus for the virus to go inside. So that way you can be confident that you can control it. So far we talked about certain viruses or bacteria which led to discovery of primarily drugs and of course vaccine. Smallpox is a kind of a viral disease caused by variola major and variola minor. It happened in historically several times, but variolation is a kind of inoculation where if you take a smallpox sore from a scratched skin and into a person, and he become infected, but his mortality will be reduced. For example, in 1776, it was noticed that milk mites, they were not affected by smallpox. Then it was found that cowpox, it's a kind of a similar virus that comes in the cow, especially in the udders. It will be there when we milk it. They will also get contracted with this kind of cowpox disease. But the cowpox is it's not a viral. You get a disease, but it's not harmful. But by the process, you get what you call immunity. So then what Dr. Edward Jenner, who is the founder of vaccinology, he took that gardener's 13-year child and infected him with the milkmaid's cowpox sore. Correct? So milkmaid get a cowpox and you get some kind of droplets or sore, and they infected the child, and the child got infection, but it's only cowpox. It will come and go. It's not like smallpox. Like the right side, you can see the photo. It's very, very, very difficult to see that. And you know that this child developed resistance. Now that way in 1800, it is a widely accepted practice. In I think somewhere in 1970, we declared that in the world it is not there. So vaccine is another kind of thing, and vaccines are developed for diphtheria, tetanus, anthrax, all these things in a way helping us to survive from all these diseases. But the important thing is that virus, bacteria, fungus, they keep on mutating themselves. They do research internally and mutate themselves and to cheat our immune system and to evade the entire human system. I should also say about a few minutes about Helicobacter pylori, which is one of the another interesting case. It has so people. It has a very interesting learning for human beings. Helicobacter pylori could be there in most of you who are attending this thing, and it may be silent in the body. The body contains every one of our body contains billions and billions of bacteria, possibly viruses, but they are silent. They live with us. And they can become hazardous, they can become harmful, notorious when they are attacked by another notorious bacteria or virus. Now they are already inside. Once this virus enters, this corona or some other virus enters and damage or start damaging all this bacteria inside, again they will take the sword and come and attack us. So we cannot say that I'm viral. Inside it's a big world for bacteria. And in 1980s it was thought that if you get an ulcer, doctor says that you have to improve your lifestyle. You are taking spicy foods, or you have a stress, so have balanced kind of life. That's what they used to say. So once you don't have a stress, you will not get an ulcer. But Dr. Marshall and Warren thought that it is nothing to do with the spicy food or with style or stress in your mind. Rather, it is due to a bacteria called Helicobacter pylori. And this bacteria is responsible for the cancer, which nobody agreed at the time when they invented. People rejected. How can they complete? See, this ulcer is the hole that is formed in your duodenal area or intestine due to the erosion. And if it continues, finally it leads to cancer. It's a horrible disease. And the doctors used to open the stomach and cut that portion of the intestine and connect it. After some time, after a few months or a couple of years, again patient will come back because it's a cancer. It is a spread to other parts. And finally, most of them died. And when they announced that it is not due to that, it is due to pylori virus, and nobody believed it. Then they wanted to prove it. They infected the animal with this Helicobacter pylori and then tried to treat and prove that. But what happened, when they put the animal, it is not able to do anything because the animal has a resistance. So they cannot induce ulcer to the animals using this bacterium. So they could not do animal test. Some people, having known that, they came and unofficially got treated. Now, unless it is proved, you cannot make it as a drug. They developed a system of drug discovery and development, and they got some grant, and they got 100 patients, and who are already infected by the disease, and then analyzed, and they proved that, and they wanted to prove that all of them are having the Helicobacter pylori and also ulcer. So that is proved. At least there is a connection. If whoever is having an ulcer, they have this infection. So first 30 patients, they found that it is not present. So they were all highly disappointed. And whereas the 31st patient, they started knowing that they are infected by this virus. Because how do they test the virus? They take the sample from the intestine and then culture it. So the normal that time practice was they do culture for 2 days, and if nothing grows, they'll throw it. And regularly they were throwing it because every 2 days or 3 days it was not there. Whereas that particular week, The technicians are engaged in something else, and for a week they didn't notice. After the next Monday they came, they found that there is a growth of this bacteria. I mean, they identified it, and then they found that it is a slow growing bacteria. Then they tested for the first 30 patients also, it was there. But even that was not published, because they thought that, okay, ulcer patients are having a pylori, does not mean that pylori is responsible for this disease. Then what they did, no? They took a broth from a patient and then they drank it. Why? To induce ulcer. Before that, they proved there is no ulcer, and they checked in the proved in the body that there is no this bacteria. Now they drank it. They want to develop ulcer sometime in 1 year or so, and then take the antibiotic and cure and show to the world. But within 3 days, he started getting, nausea, vomiting, bad smells from the body. His mother told that you have something wrong with you. His wife started shouting at us, because if he dies, what will do? Then after every day he was scanning it, then he proved that bacteria was inside, and vomiting was there, and proved that ulcer already started within 7 days. Then they took that antibiotic, which is already known, and they cured of the disease. Ulcer, both were cured. So they were awarded Nobel Prize, and those periods, it was a horrible period. Now we don't even go to hospital. We can take a tablet, call a doctor. Doctor says you take it, you are cured, and they were given Nobel Prize. So pylori gave a knowledge. Yeah, thank you. Now, all the things
please unmute your audio and video, please. Senthil, sorry, please go.
That's fine. It's okay. Once in a while it will happen. So at least we know that people are watching. So next we talk about drug discovery. So all the thing what we need a drug. So even though the microbes are doing research, we also keep doing, otherwise the human race will disappear in no time. So what is a drug discovery? I'll just give a short explanation about what it is. See, treatment is what? So you have a disease, like diabetes, you have a psychotropic. So, and you have, body has a lot of proteins, lot of targets, and you make something which goes and stops it. So the molecule, something is a molecule, small molecule or bigger molecule. If it inhibits it, if it binds to that, and the disease created by that protein will get stopped. Like that is what. So you have to identify a right target in the body. So there is a gene target mapping is there. And so if you inhibit that target, whether it gives the desired effect, so then validate it. Once the target is ready, like biologists will do the work on this line. They identify the target, like for diabetes, you have to inhibit dipeptidase inhibitor 4. For example, for each disease has its own targets, like they have a AC converting enzymes are there. So body has a lot of enzymes. If you inhibit some enzyme, and the problem due to the enzyme will be stopped, or in some cases, if you activate the enzyme, you will get a desired activity. So once you have the protein ready to be tested, then you have to make the drugs. How do you make the drugs? You have a natural product and slightly modify, and from that you can make some design. We have a computer-rated drug design. You can use the bioinformatics, makes molecules on the screen, and then try to synthesize and then give the molecule for testing by biologists. Or you can use combinatorial based, like have thousands of molecules in hand. Or there is also analog based. Already there is a drug, like a penicillin already came. Slightly I modify and see whether it is working or not. Like that. So chemists, these are all jobs of chemists. Chemists can do all these things and synthesis in the lab and give for biologists. They will check, they will come and say whether it is active or not, how much is active. And then once, after thousands of molecules, if we found that some molecules show some activity, we call it as a hit. Then optimize the hit. Change the structure slightly and slightly and slightly. Every day we keep on checking the activity and optimize it. You get a better activity, so 90% activity, 95% activity got it. Few molecules, out of the select one candidate for doing a study, clinical study. And what do? You infect the animal. Now so far you have done with an in vitro, in the lab testing. Now you go to animals. Infect animals with the disease. For example, you are developing an anti-diabetic, you take an animal, give some chemical, it will get a diabetes, and then give your lead, your candidate, and see whether it is cured or not. Once it is cured, what will do? It should not have a side effect. So give the 10 times of the dose to healthy animals, male and female animals, and give for about 28 days or 14 days. sacrifice animal and check whether it is kidney is affected or heart is affected, liver is affected, blood profile. So that way you have to have a good active molecule. At the same time, this molecule should do only that job. It should not react with any of the proteins or enzymes in the body. How do you know that? By testing its toxicity. That's what they call it as a safety. First is called efficacy, how efficacy it is. Second is safety. Safety and efficacy, these two are the two important things for a drug discovery, for a drug to be checked. And then once it is proven animals, then you go for a approval from the government. Plus every stage you should go to the government. There is government means it's not our typical government. There is a separate bodies with all scientists will be sitting there. They will review and approve you. They will approve your phase 1 clinical. Means what? Take a set of healthy volunteers, male and female, and give this lowest level of dosage and keep on checking their blood profile, organs, their performance, and then prove that healthy volunteers, it is not having any safety issue. It is not toxic to human beings. Once that is proven, then go for a phase 2A studies where you go to the patients. About 20-30 patients, you give them and see whether it is curing and see whether they don't have any toxic issues. Then go to phase 2B, take about 100 to 300 patients and do the same thing and prove that it cures the disease and it does not induce any other toxic issues. Then go for phase 3 where thousands to 3,000 patients are checked and prove that once it is done on thousands of patients, finally it is approved. Then go for a. So this is a simple thing I have presented differently. First is early discovery, drug discovery stage in the lab is tested. Then it is done in animals. Then it is a human volunteers, phase 1, 20 to 80 patients. Then phase 2, it is done on 100 to 300 patients here. Both efficacy and safety are tested. Phase 3 is nothing but of a broader population and get an approval. After approval, you launch the market and keep on monitoring the patients. If there any issue comes from the market itself. So normally it takes, the entire process takes about 14 years or 15 years, sometimes 18 years. If you're lucky, it will be 10 years, but it's a long period. So if you start drug discovery for corona today, minimum 10 years, you will not get a drug into the market. Not only that, expense will be at least between 5,000 to 10,000 crores of Indian rupees. It's very difficult for any Indian company to invest that money risk because it can fail even here or here. After launching also it can fail. Typical case of thalidomide, there are many drugs. So no company can withstand 1,000 crores of loss, even 10,000 crores of loss. So Indian companies normally do up to phase 2 up to this stage and now license to a multinational company who invests hundreds and thousands of crores to bring the drug into the market. See, in the first stage, about 5% drugs will pass approximately. In the second, only 2%. If you take 100, only 2% will pass through. Here, only 20. So overall, 1,000 to 5,000 molecules are required to be synthesized and screened to bring one drug to the market. Why? Because it has undergoes, it should pass through all these things. It should be selective, it should be stable, it should be soluble. Because you cannot have a tablet, you eat it, it's a water in your stomach. If it is not water soluble, it will go out of the body when you go to the toilet. So it should be soluble. If it is too much ionic, it cannot enter into a cell from the stomach or from intestine. From intestine, which is highly aqueous, it should go to the cell which is organic. So cells are having a lot of lipids, no? Long chain carbon CH2s are there. To enter into cell, it should have some kind of hydrophobicity. So drug has to have a balanced hydrophobicity and hydrophobicity. And third is metabolism. Liver destroys the molecule which is not of our not of its interest. So it should escape from the liver before it is active. It should not induce some other activity. Once drug's job is there, it should come out of the body. Drug should not sit in the body. Then over a period, you become a diseased person. Your blood will become impure. So once it is job is over, you take an antibiotic, job is over, it should come out of the body. It should be durable. It should not induce deformities of your child. There are drugs. It should not induce mutations. So next generation should not be affected. No, it's possible some drugs induce the mutagenic. And what happens? Children are born with deformities or some problems in the body. Should be patentable. It should be manufactured. Some drugs could not be. Why 1928 Fleming is invented it, but it took 14 years for the drug to come to market because it was not manufacturable. In fact, 1931, Fleming almost dropped it and came out of it. So I have seen it in the lab, but I cannot manufacture it. And then Florine Chain did some work, another 10 years or 12 years, and because of which it came to the market. Now let's talk about something about, now we talked about discovery, how difficult, how costly, how much time consuming. I think you could have got some idea. You can ask questions now or later also. Now, what is COVID? No, it's actually a disease that is caused by SARS-CoV virus, which is almost how many years old? More than 40 or 50 years old. It's a family of virus, out of which one became very serious, and we are into trouble today. And it was first identified in China. All of us are aware because we have enough knowledge because of this COVID. People know a lot of medical terms today, and everybody is following in the world. About 700 crores of people are following what is happening every day. First reported in December 31st to WHO, and WHO declared it as an outbreak as global health emergency in 2020, January 30, and global pandemic. It has been announced on March 11th. And as it says very clearly, unless there is no treatment, there is no prevention today, there is no drugs, there is no biologics, but you can prevent by only 3 ways of entry into the body: nose, mouth, and eyes. It is in your eyes, NME, and your hands. So these 4 are the things. If we don't allow this hand to be in touch with any of these things without washing, you are 100% safe. And these things now pass through our, once you inhale it passes through our airways. On the sides, the cells may contain the protein called ACE2 enzymes, and it easily attaches through its corona size. Corona means what? Crowns. You see the spike proteins. I think it's here. Yeah, you can see the spikes on outside this corona, and these are our proteins. This is an ACE inhibitor in your cell. This is the cell wall. This is cell wall, and cell wall contains ACE, angiotensin converting enzymes. This corona spike proteins are really used to open the key. They attaches, and once it attaches, it enters into the human cell. Inside, you can see RNA. It's typically an RNA virus, which means it cannot reproduce on its own. It needs a host cells, is a manufacturing machinery to produce itself. Once it enters into the cell, it merges and opens up. The outside is the lipid membrane. Sorry. It's a kind of a lipid membrane, fatty kind of membrane. And that's why when you wash with soap, it dissolves. And this is, once it's open up, it comes out. It releases its RNA into the cell. Cell has a machinery. It picks up that RNA. RNA is typically RNA is typically a technology. RNA has a technology how to manufacture its next generation, whereas our cell has a machinery to produce it. You can see here, RNA has what? It has a protein. It has a corona kind of spike proteins. It has also RNA. So some of the RNA are reserved separately to give to the next generation, and some RNAs are utilized to produce these proteins which are required. So our machinery, see, our double-layered membranes produce all individually. Separately, they make spike proteins, this RNA and lipid membranes, and finally they assemble it to make it. See, finally RNA is put inside. This white color is all our cell machinery. So individual parts of the virus are produced by our cell separately. You can see here, and finally they are assembled by our cell. Our cell assembles finally. Finally, this RNA is put inside and closed. This job is done by our cell. So yeah, now they are all assembling here all the produced enzymes. Sorry, corona viruses assemble in a place in our membrane, and then they break away and come in outside the cell. Like this, see, they're breaking our membrane, and all these newly manufactured viruses are coming out of the cell, and again cell is closed, and this cell, our cell, can make millions of viruses. Tons, hundreds of thousands of viruses will be made by a single cell of a human cell. Finally, it destroys itself. So what happens? Virus enters inside, it releases its RNA. Our cell machinery, cytoplasm area, our machinery produces all small individual parts of the virus and assembles it and makes hundreds or thousands of viruses and sends outside, at that point it also dies. And these millions of viruses enter into other cells, and again millions becomes millions of millions, and then millions of viruses, and all debris and fluids accumulate in your lungs, and you are not able to breathe, and you get an highly inflammation. Why inflammation happens? Once these things are happening, your immune system is afraid of something is happening there. Go and attack. So it produces what is called interleukins, cytokines. These cytokines are sent in a big way. That's called a storm, they call it. All the cytokines come and try to attack these viruses, but in the process, what happens? Inflammation happens in your airways, and airways become smaller and smaller, and the patient is not able to breathe, and finally it leads to the death. Today, what kind of treatments are available? We have only experimental treatments. Doctors examine and try with every patient what can be done. There is some protocol given by WHO, and it's also given by your ICMR. And doctors also look into the other problems with the patient. He may have a heart issue, he may have other comorbidity. So all individuals are not same. So it goes by experimental. Doctors are giving only supportive. So they want to make the person get some support. For example, if you find it difficult to breathe, put an oxygen so that he can survive for some more time. But during that period, immunity can pick up and be able to destroy virus. And so actually it's a supportive treatment. And only solution today is prevention. Like social distancing, masks, and not touching your 3 important entry gates with your fingers or with others' fingers also. There is no proven treatments. Currently it's a critical unmet need. There is only 2 types of treatments: do an inpatient care and do as much possible randomized trials to understand and treat this coronavirus pandemic. Good part of this virus is that even though it can spread very fast, 80% of the people stay in stage 1. They get only early pulmonary effects, and 20% proceed to the late pulmonary effects. Pulmonary means lungs, and out of the 20, about 20% go to the hyperinflammation, which is actually severe. Only 2 types of treatments are available: either prolong the disease and rescue those who have become severe. And the good part of it is that many persons are asymptomatic. Means, what does it mean? Virus entered and their immunity system is attacking, and even they are ruling the virus. Virus still is struggling, not himself. Since virus is in very low number, they are not able to overtake our immunity system, then it will be asymptomatic. So in general, spreading percentage from an asymptomatic person will be very less, because his percentage what he's putting outside will be very less. This virus, once it comes out of the cells, it attacks other cells. Then after that, what happens? Then it leaves the body also to attack others. Then what kind of treatments are available? First thing is that we have people give a lot of azithromycin, which is not an advisable thing, because azithromycin does not cure a viral disease. It's actually an antibacterial. But once you hit a virus infection, people become very weak, and what can happen is this: people will be attacked by the bacteria too. So in those cases, doctors should judge and give the antibacterial like azithromycin. Otherwise, 2 things will happen: resistance to azithromycin can happen, so that becomes a useless drug, and we cannot overload the patient with too much. Then he himself will be struggling to come out of that. Of course, in second kind of thing, the treatment is blood thinners, where it was found that patients who are in serious stages or who have gone to death, they were found to have some blood clots. So worldwide consensus is that if we give a blood thinners, then blood clotting formation will be less. So in a way, it's kind of a supporting. So this is also another supporting treatment. And BCG, polio, MMR vaccines, it was thought at the time. Even some of the world leaders announced that it's a wonder drug. But when the trials were done, systematic trials by UK, that's called recovery studies, and also solidarity study. These 2 studies, big studies involving thousands and thousands of patients, it was proven that it is inappropriate. It's not helping anyway. Earlier, when India was not getting too much of virus, it was thought that it is due to BCG, but today we are also getting it. So BCG is good for that particular microorganisms, but may not be for corona. And people are giving vitamin C, vitamin D, zinc. There is no proven thing, but it's always good to have such kind of thing because this is important. So far we discussed about what kind of treatments are available today and how difficult, how impossible today if you look for a new drug discovery. So are we going to stop it? No, that drug discovery will be there. But today I cannot wait for 10 years. By the time it will be a big issue. Society will have a big issue. Already is struggling. Countries are struggling. So what is the way? Can we start from phase 2 or phase 3 instead of doing early studies? How is it possible? Let us take a proven drug, the drug which is already in the market. Which means if it is already in the market, that means safety is already proven. So now we should only check whether it is active against corona or not. If it is active, then we can go because safety issue will not be there because already existing drug, and that is what is called repurposing research. The drug which was originally having some purpose to treat some disease, now repurposed to this corona disease. One of the successful thing is your steroid, dexamethasone. This is the only drug today which have a striking impact on mortality. No other drug can save from death. It is normally used, prescribed for anti-inflammatory and anti-immune drugs. It's a cheap drug. It is introduced about 50, 60 years back. Here, normally what happens in our body? Some of the people, once the virus enters, an immunity system, we have both two immunity systems. You have innate immune system, there's a genetic immune system, and adaptive immune system. That means which learned in your lifetime. You live in the most hygiene condition, sterile condition. Your immune system will not learn about immunity. The person who is living in less immune systems, his immunity will be stronger because the system would have seen many bacteria and virus and learned how to attack them. So this is what is called adaptive immune system. So your immune system is afraid and sends a lot of people there, like what we are sending to Ladakh kind of thing. Instead of sending sufficient amount of cytokines, interleukins, sends a lot of things. And what happens? So much of accumulated, these kind of cytokines are accumulated in the lungs, and it finally lead to what? Airways getting blocked. It becomes inflamed. Your airway will become smaller, and you have a breathing difficulty. It's a kind of autoimmunity. It's our own immune system. Because it does an overreaction, it leads to the death. So in those cases, if we give dexamethasone, it reduces your inflammation. It's actually it suppresses immune system to some extent. And during this period, your inflammation decreases, and person is able to breathe well. He is able to use the oxygen that is given, and during that period, body helps the develop immunity. But you should be very careful because if you take too much of dexamethasone, it completely suppresses your immune system so that you will not be able to fight at all, then it could be dangerous. So there is a balance which doctor knows how much to be given. And they did a recovery trial in the UK. It's on randomized, thousands of people are involved. About 2,000 patients were given dexamethasone and 4,000 were not given. And in those where it is given and those who were in ventilators, 2/3 were saved. And who are in oxygen, 80% of the people were saved when dexamethasone given. So that's a proven drug for treatment of seriously ill patients. This is another steroid which is under phase 3 trial. They want to give us an inhalation. Do you know advantage of inhalation? You want to give the drug to the lungs. But what do? You inject it or give a tablet. The minute when you give a tablet, only part of it will be absorbed by your bloodstream. Rest will go out of the body. And that will be distributed into the entire body of 70 kg, maybe 7 liters of blood, whatever it is. And very small percentage population, maybe parts per million may go to the lungs. But if we take an inhalation, no, straight away it goes to the lungs. Even 1/100th of dosage should be sufficient if it is given as an aerosol, because it directly goes to the lungs, and that is where you need the drug, but not in the rest of the body. So, and clinical trial phase 3 is going on. If it comes, it will be really wonderful drug. It's a glucocorticoid, already used in where? Asthma and allergic rhinitis. Remdesivir is also an interesting drug introduced by Gilead Sciences. It is also used. Its very purpose is to improve the rate of recovery, not save from the death. If you have to save for 10 days, maybe about 5 to 6 days you can get recovery, you can come back home. And it should be used only by the doctors. And how it does is, it blocks the replication of the virus. What is important in a virus? The RNA polymerase. And that has the technology how to make another virus. This molecule, remdesivir, goes and binds to that virus. Once it binds, that polymerase cannot act what it wants to do. Its property will change because this fellow is not leaving. It holds very tightly. Then this replication of genetic material will not happen. And finally, new viruses will not come. And that way it can reduce the, stay at the hospital. So new virus replication will be stopped. And if you can see, remdesivir is only up to this part. Rest is all what? Prodrug. Because it has to enter into the cell, otherwise it cannot enter into the cell. And second is your similar drug. No? See, you can see one more thing. This is typically similar to nucleic acid. You can see the sugar part, and you can see this. Whereas sugar, we have this pyrimidine. Instead of nitrogen here, you have nitrogen here and here. So this is to cheat that virus polymerase. It looks like a nucleic acid, like adenosine or guanine, but it is not really so. So. And it thinks that this virus polymerase thinks that it is also in another nucleic acid. It goes and binds, but in reality it's not a nucleic acid. So this polymerase got cheated, but this drug does not want to leave it. So finally replication stops. And going forward, we have a similar drug. Similar to that, you can also see the almost similar to remdesivir. You have a sugar part, you have nucleic acid. Of course, it's like urea. Uracil is a similar molecule, and that is under now phase 2. And Ritback for biotherapeutics wants to give to for the country, for the world. So that is really a good contribution, and still it is under phase 2 is going on. It is also similar to our remdesivir in terms of stopping the copying errors. Like it cannot produce new virus. And then going to this is one set of molecules. You took a nucleic acid analogs to stop the replication of the virus is one thing. Second is monoclonal antibodies. You can see the last map, MAB means monoclonal antibodies. All maps are monoclonal antibodies. Not why they call it as humanized. Actually, it is produced in the animals. They give similar vaccines to animals. Animals produce antibodies, and they did some cloning to make it similar to human acceptable antibody, and they call it as humanized monoclonal antibody. It is used originally for rheumatoid arthritis. Rheumatoid arthritis is what? It's an autoimmune disease, and in our case also similar to autoimmune only. So then they want to, and this immunosuppressive drugs, it suppresses immunity. That's what we want, because our immunity is overactive. So we want to partly suppress it, so that inflammation is reduced and the storm of cyclic cytokines are stopped. And that is also doing well. But here, this is authorized for emergency use, which means there is no complete evidence. But today we don't have anything. So we want to save the patient who is dying it. So agencies, Drug Controller General of India has authorized for only emergency use. Even Australasian Society also given for emergency use, because during cancer treatment, you need this kind of immunosuppressants, because there will be a lot of cytokines sent by immunity, and we want to control it. It is already used in cancer treatment, and that is even can be given here. And itolizumab, it has raised big criticism across the country. This is a Biocon, it's a innovator. It was originally approved for chronic skin disease called psoriasis. You get a patches on the skin, especially on the knees and elbows. You would have seen that. And this is an can be given for autoimmune disease. Itolizumab is already in the market from 2013. And Biocon did a very, very small study. They took only 30 patients. 20 patients were given the treatment and 10 patients without medicine. And the very, very small sample size. And all the 20 who were given this itolizumab got cured. Whereas those 10 patients who were not given, 3 died. So they thought it is doing well. Drug Controller General of India has exempted from doing phase 3, which is never in history, or never it is done. At least they'll do at least few hundred patients, they will do it. That's why it is drawn criticism. But since it's already used for psoriasis from last 6-7 years, drug controller could have exempted it. This is a cost. Cost per treatment is about 32,000. It is a new monoclonal antibody. It's also, you can see here, MAB. It's a monoclonal antibody which is approved a couple of days back, somewhere in July 14th or 15th it is approved. And then you have another famous drug which is approved by Drug Controller General of India, but not at US or Europe. Please keep that in mind. It's a favipiravir. Indian companies can manufacture very, very simple molecule. You can see this is a pyrazine, one fluorine, one hydroxy group, and an amide group. Very small molecule, and it is approved in Japan in 2014 for influenza. Now they are trying to repurpose it. Even today, US and Europe has not approved it. Even the phase 3 is completed. You know why? This can induce teratogenicity in animals, and it is expected it may happen in humans. Teratogenicity means what? If the pregnant ladies take this drug, the children will definitely will be born without limbs or no hands. So deformed or unformed, malformed limbs or hands will be there. It's very dangerous. You can think about the thalidomide case which happened many decades back. It is a drug. It's actually a prodrug. Once it is given into the body, it forms favipiravir ribofluoronoside 5-triphosphate, which can be given orally or injectable, and it gets converted into favipiravir in the body. Japan has approved it under conditions, and it also inhibits, similar to our remdesivir. This is also inhibiting the polymerase. So it binds the polymerase. Polymerase is having a technology to produce the next generation. Since it is binding, it's not active. Yeah, that's fine. It's also found to be active against Ebola virus, which is more dangerous because 40% of the people who are contracted Ebola virus will die. Unlike your corona, it's hardly 3% worldwide. Maybe in some cases, some countries 5% or more than that. But in India, it is about maybe around 1.5 to 2%, or maximum it can go to 3%. And West Nile virus, yellow fever, there are many viruses it is active. Yeah, and this is another very famous drug called hydroxychloroquine sulfate. It got the prime name by President Trump, who said that it's going to be a wonder drug few months back, but it is not really. So originally it's an anti-malarial drug. Later it was already repurposed in 1990s to rheumatism. Last 25 years, it is marketed for both malaria and also rheumatism. Now it's thought to be for having activity for against SARS-CoV-2, but it's not really. So lopinavir is another antiviral drug. It is active against human papillomavirus, but it is proved to be not. Actually, ritonavir is another drug which actually inhibits the protease inhibitors. The enzyme that metabolizes the protease. See, protease inhibitors in the virus should be free, for the protein to come out. Proteolysis has to happen. Then only protein will come. So once protein is produced in the body, in our cell, that should be cleaved and then put into the virus. But this inhibits the protease inhibitors, protease enzymes, and the protein will not be able to come out. So manufacturing industry of our cell cannot give the proteins to the virus. So that way ritonavir acts. It works on hepatitis C virus and similar viruses, and it is approved for HIV and AIDS in 1996. It has a different story in itself, and then people tried both in a big study, clinical study. Hydroxychloroquine was also tried in a big study, WHO's solidarity study, and the United Kingdom's recovery trial. These are all thousands of patients are involved to know whether it is working or not. Then they proved that both of them did not work. Hydroxychloroquine sulfate did not work, and even lopinavir, ritonavir also, which is known to be active in another virus, is not active here. And the FDA has revoked the emergency use authorization. Initially, it has given it due to pressure, but it has revoked the emergency use also today. WHO also stopped further trials. Why it is not active when it works in several cases? Why it's not active against in actual study? They did some study. The scientists from University of Basel and in the hospital of the university found that the concentration of these two drugs in the lungs, in the location where your virus is, there is very less. You take orally or injectable. So what happens if you take an orally? It goes to the blood, or if you inject it straight away, it is in the blood. But it has to go to the lungs. So the concentration of these compounds in the bloodstream is very high, whereas if you go to the lung compartment, it is very low. So if it cannot go to the lung, if drug is not delivered at the required site of action, it's as good as inactive. So when you do outside in the flask or test tube, it will work. But if you take it, it will not work. But in my opinion, I believe that if you can make an aerosol, if you can deliver it through inhalation, straight away it will go to the lungs, it may work. But as on today, taking as a tablet or a capsule or an injectable, it's proved to be not working. Then another fellow, so far we have seen those kind of drugs, no, antiviral drugs, and what is the autoimmunity, monoclonal antibodies. And also we have seen immunity kind of drink, immunosuppressant drugs. Now we see anti-parasitic drugs. Ivermectin does wonder outside in the flask, in a cell culture to kill the coronavirus. But in actual trials, it is not working. And they tried ivermectin with bicalutamide. It is studies going on, phase 2 study. Then you have a nitazoxanide. It is also under phase 3 trial. This is also another parasitic drug, as well as it is antiviral, helminths for protozoal. It's already active against helminths, protozoas, and virus. And that study of phase 3 is under trial here. And you have umifenovir. It's a viral drug. It is not even approved for influenza in the United States, but it's already approved in Russia and China. And they use with caution statement has been given. You have baloxavir marboxil. This is also approved in the last year in Japan for influenza virus, and it is under study phase developmental studies. You have baricitinib. You can also see here. You can see a typical adenine kind of base. Of course, there is some changes of the structure to cheat your virus. And along with remdesivir, this is being tried in clinical phase 3. And you have a ruxolitinib. This is analogous to our earlier drug, which is also under study for those with respiratory distress, difficult to breathe, and who are in ventilation, mechanical ventilation. And you have a tofacitinib, which is having a similar group, variation of the earlier one, which is currently used, rheumatoid arthritis, which is an autoimmunity disease, psoriatic arthritis. So it is also known to suppress your immunity. That way, phase 2 study is going on. You have a ribavirin. This is, you can see, it's a still modified version of base, and you have a sugar unit. This is also under drugs. You can see the more than 2 dozens of existing drugs on our trial, and each has a different categories. These are nucleotide drugs. Earlier I have seen steroid drugs, you have monoclonal antibody trials. Now this is a kind of a nucleoside drugs, which are also being investigated. And now we come a different category. You take an anti-inflammatory drug called imatinib. These are all NIPs. These are all anti-cancer drugs. NIB means it's a typical anti-cancer drugs. So basically that is to suppress your inflammation, which is there in the lungs, which is the responsibility for your difficulty to breathe. This is under phase 3 study. And you have another, along with the dexamethasone, which is currently used, they want to put on antiviral drug, atazanavir, which is currently used for AIDS and HIV. And this dexamethasone used in COPD, chronic obstructive pulmonary disease, and you have rheumatism, skin diseases. So this combination is under trial now. And you have a losartan. It's a cardiovascular drug. AC inhibitors. This is a typical AC inhibitor, angiotensin converting enzyme inhibitors, currently used for high BP, diabetic kidney, heart failure, and all. And they say the hypoxia, when oxygen level is low in the lungs, whether they can help us, because AC inhibitors are there across the lungs, that can help us to enlarge your ventricles and all. And you have a duvelisib, and this is a little different mechanism. So it works on your hyperactivation of innate immune system. Innate means your genetic thing, and it can polarize. See, as soon as the first innate immune system, if it sees a virus like an antigen, immediately sends a lot of macrophages. Macrophages are what? Big proteins. This is a big bag. They put all the proteins, close it, and through your system, body, lymphatic system, it tries to throw it outside. So, and if too much of macrophages goes into your pulmonary area, you get into difficulty in breathing. So, to suppress it, this kind of anti-immune suppressants can be used. And the study is going on. You have prazosin, you have apilimode. This is another kind of thing which inhibits the production of cytokines itself. and sirolimus, that's also under phase 2 study. You can see chemists have a very, very big role. All are small molecules made by chemists, designed by chemists. All the drugs were already brought to the market by chemists and in collaboration with biologists and pharmacologists, and that is the reason today we have a big array of existing drugs that could be repurposed. You have a chemostat, you have an naltrexone, you have a ketamine very calculated, even naltrexone. Both are what? They're opiate addiction drugs, alcoholism drugs. This is also known to suppress your IL-6. This is ketamine, which is used in anesthesia, even commonly used. If you give a very, very low dose compared to anesthesia, don't go to anesthesia dose, there it is known to rapidly reduce the production of these cytokines, inflammatory compounds. If inflammatory cytokines are reduced, your inflammation in the lungs will be reduced, and you have more chances to breathe and come out. You have a dipyromidine, it's a different category again. It's a nucleoside transport inhibitor. So it will not allow to transport from one place to it binds to the thing, so protein, so that they cannot transport to a required area. So it again works on a different mechanism. Indomethacin, it's a non-steroidal anti-inflammatory drug, which is also being tried along with hydroxychloroquine, leflunomide. Now, leflunomide is another drug, which is an anti-rheumatic drug. No, like this, about 25 to 30 drugs are being done. Something will come, but that even phase 3 takes us some time. It cannot come within few months, but we may have drugs maybe in 6 months period. Something will come, even if it is done at a faster speed. Now, immediately, what is there? You have a convalescent plasma therapy. Those who have been attacked by the virus, and then those who have recovered it, and they will have the antibodies in the blood, and that will not last for long. In the first month, it may come to about 50% may reduce. In the second month, maybe another 50% may reduce. Even third month, it will come very low that the person can again may get infected with the same virus, because this kind of antibodies memory is less. And these antibodies, if it is taken outside, given to the patients, there's a very high chance of success, and toxicity will not be a big question for in this case. And that should be used by a doctor with an appropriate care. That is very important. 1 second. And then going to the next thing, there is another interesting case. You have a PLX cells, that is plasma expanded cells. Before, see, you have a postnatal placenta. After the childbirth, you have the stem cells. Stem cells, they're undifferentiated cells. They can take any form. And it is developed by Pluristem company in Israel. This is going to be one of the wonderful treatments. Here, the important thing is that what normally we are getting, we get a lot of macrophages. You get an immunity. These cells, once you put into the body, they do two jobs. They reduce the immunity, and also they develop kind of antibody kind of cells to attack the bacteria, the virus. It has immunomodulatory properties. Number one, it induces the immune system's natural regulatory T cells. And second, M2 macrophages, and prevent overactivation of immune system. So these are wonderful drugs, but once it is demonstrated, it can be produced very easily, because from a single mother, from a placenta, postnatal placenta of a single mother, we can generate medicine for a lakh of people. So it's under again development. It is very important for acute respiratory distress syndrome of the patients who have gone to serious stages. And now, just looking at the vaccines as is another kind of treatments. Worldwide, people are under various phases. You can see the rabies preclinical test. That means preclinical means what? In testing in animals. Phase 1, orange is in testing on human volunteers. And third, phase 2, yellow color is phase 2 on a small group of patients. And phase 3, this is color what? Blue, on thousands of patients, and green means approved by the regulators. Every country has a drug regulator who approves each drug to use in that country. If we take China, there are a few companies. Wuhan Institute in Sinopharm, it is in phase 2. That is, few hundred people are being tested. It has passed phase 1 and phase 2. And in Butantan, in Sinovac Institute, it is in phase 3. It has crossed preclinical phase 1, phase 2. Now it is in phase 3 in thousands of volunteers. CanSino Biologically Incorporated in Beijing, it is approved now. It is in approval stage. And Beijing Institute is in phase 2 study. If you go to United States, there are two companies, Novavax and Moderna, both are in phase 2 stage. And Germany, BioNTech in phase 2. And United Kingdom, Imperial is in phase 2, and you have AstraZeneca, very famous. You have been seeing in news now. AstraZeneca is a company, Oxford University, in collaboration. They're in almost they completed phase 3. It is being reviewed in parallel. In Australia, you have Vaccine Private Limited, which is in phase 2. In India, we have 2 companies, Zydus Cadila and Bharat Biotech. Both are in phase preclinical, and also they started dosing in healthy volunteers in phase 1. And you should know about this Oxford. What is it about? Oxford, that's very important for you to understand what it is. No? This is the first vaccine that is going to be accepted worldwide. And this is done by Oxford University, and you have AstraZeneca. By a systematic study of thousands of people are involved. Now the thing is that important thing about this vaccine is that it has dual immune response. First of all, how is it made? It is made from an adenovirus, very simple virus. What you have already seen, it is a simple cold kind of virus. You don't get anything. You may get a cold, but nothing will affect you. And they take adenovirus, which actually normally infects chimpanzee animals, and they took that adenovirus and inserted this kind of spike proteins inside, did some kind of genetic work, and inactivated that virus and put on and created a vaccine. This adenovirus will not harm us because it's a very simple virus. We may get a cold, but it will not harm us. But inside that there is a mutated protein, is the protein of the coronavirus. Once our body sees that, we will develop 2 things. Immediately our body will develop antibodies and also T cells. And it has been proven in volunteers that it does both the jobs. That is an important thing. It does not only create antibodies, because antibodies may die after a few months. That means every few months you should keep on giving vaccine, which is not going to be probably, may not be practical. And you need to give one thing like polio. You give a vaccine, it says for lifelong. And so far, we could not get a vaccine for SARS or MERS or similar coronaviruses. But in this case, which is created from an adenovirus, it also produces T cells. And what it does, these T cells remember it. What is the technology? And to produce those antibodies and how to identify. And it hunts for viruses to identify whether that fellow corona has come inside or not. If it has come inside, immediately instruct the system to produce antibodies. So this is very important, because that gives you durable protection. If you give it, it stays for long in the body. But we should also keep in mind about safety, because it is done with a selected undiverse, undiverse group of healthy individuals, not with the wider population. But if we really, and it does very well, but if we, in real world, there's a broader population. So Indians are different from Chinese, are different from Germans, are different from Negroes. Each body is different. We don't know how it's going to behave. Number one, there could be older adults. And third, maybe they have other disease, which also could be making it more complex. But these things are always going to be there, but something has to come and that is what has come through this vaccine. And let us expect some more vaccines to come in the due course. So far, to summarize, what has happened is there are some treatments available today. There are some treatments like we have dexamethasone, you have remdesivir, you have certain monoclonal antibodies, you have some more antiviral drugs, and new things are going to come, new vaccines are going to come. Currently, we have a plasma therapy, and also PLX, expanded cell therapy, is going to come. But above all, the thing is that we should now also collaborate and cooperate. First, we should help us not to allow inside us. Once it goes inside, you don't have a control. And without your activity, it will not go inside. You touch anywhere, don't worry about it. At any point, your body may get contaminated. There is no surety that it will not be there. But don't touch your fingers with either eye or nose or mouth. If it requires to touch, please wash with the soap and then do anything. And keep on washing once in a while, so that even if it is somewhere, you'll keep on washing. So to avoid accidental touching of nose or this thing, you put your mask so that accidentally you will not touch. But if you don't have, if you believe that by mistake I will touch it, please put a gloves. But once you put a gloves, you will not touch any of your face parts. Very easy to protect it yourself. And second, many of the cases, what is admitted or discharged, most of the cases, maybe 90% of people do not even require any treatment. They could have easily come out of it, even if nothing is given. But today, psychology is the doctor has to give something. Doctor also want to give something. And people lose the confidence if nothing is given. So doctors are treating. But please don't treat too much, because sometimes the Hippocrates, his ethical says, better not to do any harm. So doctors should not give too much, because sometimes giving something may create more problem. If somebody does not require, but you give a dexamethasone, his immune system will become too bad. That will be dangerous. Leave it and see if it is required and give it. So there are three prongs. One thing, you protect it. Second, only if it is required, you give a treatment. And third, don't over-treat it until you get really well. But please keep in mind, this corona is not the end of it. There are going to be more things, because this bacteria, this virus, they created this world, including you and me. The life is brought to this world by cyanobacteria and this kind of microbes, and they are more intelligent and they can bring anything at any point of time. So this 21st century is going to be a big challenge. Not only 21st, this millennium is going to be the challenge. Is life endless? The challenge for microbes, but we need the scientists. Scientists are very important. Otherwise, the race will disappear. And RJ Science Foundation, we do little, very, very small part to bring passionate scientists to the world. We take the postgraduate chemistry students, where Dr. Ponnuswamy is a founder member, and many of our friends are there, and many of our enthusiastic youngsters, seniors of RGSF for the last 9 years we are conducting. The seniors are doing a wonderful thing in not only training the students, they want to create interest in chemistry for the students. That's what they do. They don't teach it, but their first purpose is creating interest, because science is interesting. But today somehow people started feeling that subject is tough one. But it's not. Everything is invented by human beings. It is not given by somebody. If it's invented by a human being, it should be interesting, because this can save our life. If something can save our life, that will be interesting. So first part, students would have got some feeling of toughness about some chapters. So they are running away. We call the people and bring wonderful teachers, extraordinary teachers, to just make the boys to enjoy learning, and also motivate and mentor them with our team of RGSF seniors and members of RGSF. They work together, and with the wonderful teachers with us, we create a passion for research for the students. And our aim is to create scientists who can do great things for the society after decades from now. And many of our friends are there. Let me thank all the RGSF friends and RGSF seniors who are actively doing last 2.5 months. They are doing daily classes are going on. And these people are doing a mentoring of 10 to 12 students each. And those activities are having a big vision. And we are traveling to the vision, and many people are part of it. Let me thank RJSF. Let me thank Professor R. Jayaraman, who's inspiration for us, and all my friends who has done PhD along with us. And also let me thank all the students. And this is a challenge for the chemists primarily. Chemists can bring new drugs. And please take a note, there is a big opportunity, and biggest opportunity and the largest opportunity for every chemist. Please enter into it. It will be very interesting. All the best and success. If you have any questions, please ask any questions you have now. Thank you.
Thank you, Senthil. Thank you for the wonderful and highly informative session. You took us to the over the years and different pandemics, and really interesting, highly informative. And it is tough to get all those informations in one hour. Very informative. I think all the participants have given a very good comments, because they said it's a very rare and highly useful session. Now I hand over the session to Dr. Velmani, sir. He will coordinate the question and answer session.
Thank you. Thank you. Sir.
Thank you, sir.
Thank you, Ponnuswamy sir, and of course, thank you, Senthil Kumar sir, for the great informative session. In fact, it is useful for all of us. And we'll have some questions for you. Sir. There are a lot of questions here.
Sir, can you, Senthil, sir, Do you ask them to unmute and, or you read it from chat box, sir?
Yeah, I shall read it from the chat box itself.
Okay, sir. Okay. We also request the YouTube viewers to put your questions in the chat box.
Right.
Dr. Velmani sir will present your questions.
Yes, sir. Sir, probably I'll come from the bottom part. So here, a few questions are here, sir.
Yeah,
Yeah, let me read it one by one. Sir, there's one question from Sai Kritika. She has given us, sir, you might have heard that these viruses are man-made. Are man-made viruses possible?
yeah, so good question from the layman to the student to the scientist to the topmost administrators of the world. China only knows whether it is natural or man-made, and after about a few years only information will come out. But it is possible. It's all about mutations. Mutations you can do at lab. Even the vaccines what we are creating now, the Oxford University along with the AstraZeneca, it is made by mutations, genetic mutations only. So genetic mutations can be done in the lab. And I want to tell you one thing that is very serious thing. Like polio virus, we eradicated completely. And some of the viruses for which we have created vaccines and completely eradicated, they are still stored in some labs. It's very dangerous. Why they are stored? Why they are stored? Because basically their worry is that at some point in future we have to develop some kind of vaccine. We need the standards. But it is possible that if somebody puts outside, it's going to be very dangerous for the world. And war, it's not a big thing for us between country to country. War with this bacteria and virus is going to be dangerous. And these mutated versions can be made naturally because body also does a mutation. You have seen that there are 8 mutations. Even if China has made 1 mutation, other 7 are made by human beings. Our cells mutated. In the body, virus get mutated. And the about truth, we do not know. Whether it is made by them, there is a scientist who keeps saying that it is done by them, and many people is not alive today. A lot of things are being heard, and some very responsible persons also say, scientists say that it is made in the China lab, but it is not avoidable. Like we can't avoid it, but we can only prevent it from entering to us. So there is a science by somebody say mutated it. The science should equally grow in this country like ours, and unless we improve, this country has good infrastructure and scientists to combat, it will be very difficult in future to handle it. So India has a 20% of the population in the world, and the highest density. China, we could be second to China in population, but in density we are more than the China, and we have very high responsibility to save our people. So we should come forward, and somebody is challenging us through that genetic or mutations and putting in the market through some purpose, some political or economic purpose. We should be able to save through the same science.
That's fine, sir.
Thank you.
Sir, the next question is from N. Jivanandam. Oh, yeah.
Yeah.
Oh, sorry. Yes. Yeah. Correct. Yeah.
Sir, நீங்க போடுங்க.
The Senthil sir.
Okay.
Velmani sir, நீங்க இப்ப வீடியோ ஆன் பண்ணுங்க. Velmani sir, please use.
இல்ல நான் ஆன் பண்ணிருந்தேன். I thought he was calling.
ஆன் பண்ணுங்க.
Okay, fine. Sir, there is one question from N. Jivanandam. Why do COVID-19 virus attack the lungs very effective? Maybe a short answer will be enough.
Yeah, actually it is going everywhere, but other places it is not creating a problem because there is a airway is there in the lungs. There are small air pockets. Other airways are there. When it goes there, it goes to every place. But there inflammation happens, and inflammation happens, the airway becomes smaller. And other organs except heart and lungs, even other organs do not work. We will live for some time. Even kidney does not work. Many parts does not work. Even brain does not work. we live for some time. Whereas if lungs does not work, we die very fast, even heart and lungs. That way, if the airway becomes smaller, breathing becomes dangerous and we die immediately. Diseases are same, but there it is more critical. Yeah.
That is another question from Dr. N. Kritika. So, what is your opinion on plant-derived antiviral drugs? Ayush has recommended Kumar's and is being administered to COVID patients.
Yeah, really. In fact, I have been taking often. I take 3 days. In fact, I recommend because we are working in our company, 1,000 people are working, and a few people keep on getting it. I'm eating in our canteen. Many people are eating there. Even canteen employees also got it, but they are also cured. But in our company, very few people are getting it because we give Kumar's in our canteen. Everybody takes it.
Right.
We ensure that people are not affected, and I'm taking it. So please take it. And today, the wonderful medicine we have in Siddha and Ayurvedic and our natural things, that is the best cure for us. And we have something. Why should we look for some other medicine? We have proven thing. Please, every one of you should take it. And there are a lot of things talked about Siddha and things. Please take it. There are proven drugs for thousands of years. And now we are looking for the drug which is not even invented.
Okay.
Please take it and you'll be safe. Yeah.
So there's 2 questions from Arul Prakash. He has made a distinction between, as per World Health Organization, initially they said it is a virus. Later they said it is a bacteria. Whether COVID is a virus or a bacteria. It's a very simple thing. I get confused. It's virus.
Yeah, it's a
virus. Name goes like that.
Virus is, coronavirus. SARS virus only. It's very small. If you compare the size of the cell and virus, it's very small. Yeah. It's only virus.
Sir,
but you know, virus enters. Yeah, please go ahead.
So, second one is, we are well aware that a vaccine needs more than 12 years, at least 6 years to market. Do you advise the present vaccine? I don't know what he is asking about.
Yes, vaccine should be required because if you ask about the usefulness of the virus, it will not take 6 years, maybe in 6 months it will be there. The latest one will come 6 months. And our Indian company, you know, our Pune company, they are going to manufacture it. They can make in billions of dosage. Yeah, Serum Institute of India, Punawala, they are going to do it. So the thing is, if you ask me, record is required. There are many things today we are surviving without disease because of vaccines. You know, in the childhood we are given several vaccines: BCG, MMR, polio. These are all very important for us. That's why we are having many things we eradicated. Whether it is there or not, vaccines are more important than even your chemical medicines. Chemical medicine comes next only. Chemical medicine, what it does, it takes care of your immunity role. Whereas vaccine, what it does, it activates your immunity system. Vaccine works with your immune system. It only activates it. Whereas chemicals takes the role of immune system. So chemicals are not really advisable compared to vaccines. Vaccines are very, very important. And that is the only way we can fight against the viruses. Yeah.
Okay, sir. And of course, there are small other questions available there, sir. So there's one thing, there is SVK Kumar. He has said it's highly informative and useful. If remdesivir controls replication, then why are we not using against COVID-19?
We are using it. It's approved in India for emergency use authorization. Doctors can use it. It's being used. It's being used in India and across the world. Favipiravir is having a problem because you cannot give to pregnant ladies. But remdesivir is already a known drug. It is being used, and that reduces the hospital stay.
Okay.
Yeah.
Then why only nose, eyes, mouth are the entry of for virus? What about other entry barriers in the body?
There are nine gateways to our body, and 5 are 2 eyes, 2 nose, 1 mouth. These 5 are often utilized. Here normally not used, but through skin it cannot go. And if you have any problems on the through skin is a wonderful protection in the body. You can even dump your virus in the skin; it cannot go inside. But if you have some cuts in the body, be careful because it's easy entry. Yeah. It's good that all the 3 are only in the face, so we can take care very well.
Yes, sir, I think this what is available. There's one more thing, sir. How long can COVID-19 stay on an object live?
It can be there forever unless it is eaten by some other
Something else.
microorganism. Because it is not a living body, it is a particle. In lab, we have, for example, sodium chloride. It is one more particle.
Yes, yes.
But something is in the environment which leaches that lipid, then it will die. Otherwise, it's one thing. Bacteria can live for hundreds of years.
Yes.
Even though it is a living body, it can form a shell around it. If there is an unformed, for example, bacteria forms an air, and in some places we are putting in anaerobic conditions without air. What it does, it will not die. It will cover its body with a shell. It will divide its body into two parts. One part it will cover small, rest part it uses as a second cover. It will be there for hundreds of years. Once comfortable condition comes in the atmosphere, it will come out. Such technology, even human beings or plants or animals are not having. So always bacteria and virus are going to rule the world. Thank you.
Okay. There's something about designing, sir. There is one by Srina Radhakrishnan. Sir, how do you think that the computer-aided drug designing can help in quickening the drug discovery process? And related to that, can you suggest some few drug designing softwares, please? This is basically much. So both combined, I've just asked you, sir.
Yeah. See, the thing is that first part is that that is what normally we do. So far conventionally we look at the known drugs or known molecules, say hundreds of molecules, and see the structure and activity. So human brain has a limitation. At a stage we cannot put 1,000 in the brain and compare their activities, their polarity, their hydrophobicity, distances. What computer does is it considers any number of molecules. You put 100,000 sticks. For each molecule, list out all the parameters, like hydrogen bonding efficiency, distance between two atoms. So like that, polarity, solubility, and activity. You compare all these things and evolve that. These are the parameters which are responsible to have a new drug. So instead of making 1,000 molecules, if we have a computer-aided drug design, you need to make only a few dozens of molecules, maybe 100 molecules. It reduces your discovery time. It reduces your efforts to make thousands of molecules and a few hundred molecules. Like that, a lot new softwares are coming. Serious2 is one of the softwares, and there are many such softwares. Then I can share with you. Serious2 is one such thing which is available today.
Okay, sir. There's one. Tell us about plasma treatment from Padmini Kumar.
Yeah, plasma is. Yeah. Good. So plasma is what? One person has got a disease and he is cured by himself. How he cured? In the body he produced antibody. So take that antibody, put it in our body. So same antibody will help us, will start killing it. By the time it kills the thing, virus becomes smaller in number. During that time, body immune system picks up, because only there are few virus, and body immunity also starts working with it. So it's a kind of helping one person with another person. Yeah.
Yes, sir. I do have my own questions, sir. Maybe the last one, I believe.
Yes.
Sir, repurposing of drugs, or whatever you say it as. Likewise, how many has been there in the market? So there's a disease there and you try to do it, and there are a lot of other medicines. So how actually they coordinate among?
Actually, repurposed drugs, there are actually hundreds of drugs in every area. Okay. Today, everybody is doing one particular thing, but even last few decades, even hydroxychloroquine was introduced for malaria. Chloroquine was the first drug. They put hydroxylation, they got hydroxychloroquine with some more change. But they found that many things are found accidentally. They found that rheumatics also is improving. So then they found that it is working in rheumatics. Good thing is repurposed is already it's in the market. We know that it is not toxic. So instead of really inventing an entirely new drug, let us screen all the known drugs. So this is what all even frontline multinational companies are doing. They take the all existing drugs and work for the new things working or not, new disease or infections. So repurposed drugs is one of the best techniques to bring the drug to the market very fast. And also these kind of things, since already proven, it will not give a new challenge to the field. So it's one of the things first we should check for any new disease before going for an ultimate new drug discovery. Yeah.
Senthil, I have one question.
Sir, one more, sir.
Sir, one more. Sir, shall I ask one question?
Yes, sir. Yes, sir.
It's a clarification, Sandil, because you said out of 4,000 drugs, only one come to the market. But you see, yesterday or day before yesterday, our TV channels are booming about Covaxin. Still, it is in the first phase. But what about the possibility or success of the Covaxin in the current scenario?
Yeah, yes. Correct. Actually, Covaxin, they started announcing only they get a success in the so the laboratory stage. So before that, they would have done a lot of things. But compared to new drugs, vaccine is a little better because they create from the virus and bacteria itself. Second is when you ask about how best or fast or how risks or danger is that it can fail also. First is it can succeed. And second is that it may not work in with all the mutations of the virus. No, there are about 8 known mutations today. The Covaxin, what is created by this our Bharat Biotech, may or may not work for all the people because Tamil Nadu, there are about few lakhs people, or would not have contracted the same virus, same types of virus. So it may or may not work. That's the first part. Second, over a period, this Covaxin, the corona also may take a new shape. It will also keep on changing its mutation. No, it keeps changing. For those viruses, this vaccine may not work. So it's all experimental. So for Indian drugs to come, it will take a long time. Before that AstraZeneca will come, Russia things will come, China things will come. So, but we should keep on searching because in the case of virus, it is not like several thousands. Whereas in small molecule drugs, we need to make thousands.
Wait, please, you leave the meeting. You have pushed the presentation button. Please, Vivek Dubey, please, you leave the meeting.
Dubey, yes, Vivek Dubey, kindly.
Yeah.
Please don't present here.
Maybe
leave, then you can come, enter.
Yeah, yes, sir. Sir, I have my own another question, sir.
Yes,
So usually, when you're trying to put into some development process, do you really expect a disease to come and then you do the drug discovery, or based on certain other factors you do the drug discovery?
We do drug discovery only after it comes, because
only
we need only when it exists. After it comes, see, many people are affected after the only discovery.
That
is one. Second, one more thing is that many of the treatments today are not really curing. They are all basically for treating the symptoms. Symptoms. Even we do not have a drug for diabetes. We do not have a drug for cardiovascular things. That's the reason that doctors give the tablet for lifelong, because every time it suppresses. So this disease is there for decades. We are still treating the symptoms. So that means there are even hundreds of drugs for which still we are treating the symptoms. So it's a very late running process. Yeah.
Oh, okay, sir.
Vivek Dubey, please leave the meeting and you come inside using the join meeting. Don't touch the present screen.
Mr. Vivek, you can go through the YouTube link. Please don't come into this thing. Sir, lots of just boring, sir. Many have given very good compliments for your speech over here, and I don't find any much of. There's one by Anantalakshmi Jayaharan. Is there any protection against rheumatoid patients who are taking chloroquine hydroxylate as disease maintenance? Probably related to this, is already being affected by certain patients.
See, today, these are all already affected cases. No, they are already affected by autoimmunity, and now they are trying to treat the symptoms only. This whatever immunosuppressants are there in allopathy, there is no complete cure. They are able to treat and manage the disease with, say, for example, dexamethasone or this kind of monoclonal antibodies. These are there. There is a treatment systems that are available, but always, in my opinion, there are certain set of natural medicines who made the people live for thousands and thousands of years. So we normally ignore after this allopathy has come. Allopathy, let it grow. We should support it. But equally, we should not forget that there is something else we had for thousands of generations. So please look, something is there, and we should always invest our time and energy and equip them to do re-investigate, validate this natural medicines, including Siddha, homeopathy, Unani. All these things should be addressed, because here we are trying to find something new, whereas there it could be already there, but we have lost over a period, because last couple of centuries we didn't give importance, we didn't give trust, we didn't give an investment to grow or train or develop those kind of existing natural medicines. Possibly some cure is there. We should all keep looking into it. Yeah.
Then question I think. Okay.
Mohanraj, it is not audible. I could not hear. There is a lot of disconnect in between.
Yeah, there is Senthil, another question. I will read it. How do you think that computer-aided drug designing can help in quickening the drug discovery process? Can you put
it really helps.
By Srinath Radhakrishnan?
Yeah, good. So computer-aided drug design, how it can speed up? So I need to make, say, thousands and thousands of molecules to find a good active hit and then lead. But I do blindly, because human brain has a limitation. If I have 100 molecules, first I make 100 molecules and generate the data. Now keeping all the 100 structures in the mind, and then all these structural properties in the mind, and all its activity values in the mind, if I try to understand something, human brain has a limitation. Whereas computer-aided drug design can store even thousands of molecules and analyze all the parameters and give you a suggestion. This structure, this group here, this polarity here can improve. So based on those parameters, if we can design, instead of making thousands of molecules, we can make only hundreds of molecules. So that is the time. How much time it will take to make 1,000 molecules? How much time it will take 100 molecules? If you use the computer-aided drug design, CAD, you need to make hardly 1/10 or 1/5 of the 1/20th of the molecules, and that saves a lot of time. That's a huge amount of time saving if we can use computer-aided drug design.
Well, Mani sir, any more questions?
Excuse me, sir. I would like to ask the question directly to the speaker.
Please,
Can
please.
I?
Please.
Dr. Senthil, this is first thing is let me congratulate the organizers and the speaker for providing a wonderful session and intensive interpretation on the drug discovery for combat COVID-19. Now, my question is, sir, we are talking about the drug repurposing. We all know that there are many advantages of drug repurposing. One could be the proven safety of the use of the drug, or it can also be the circumventing the cost and the time period of producing a new drug. Now, sir, this is a direct question to you. Now, since you are in the pharma company and an R&D scientist, you please reveal us the real advantage of going in for a drug repurposing.
Yeah, actually, you said two things. One thing is bringing fast, and then safety.
Yes.
In my opinion, bringing fast is one thing, but safety is the most important thing, because now certain areas people are pushing faster to approve the drug. Yes. Because a lot of political pressures, and they approve it. But in this process, it is possible that some drug may escape. Like one case, itolizumab, like a Biocon's drug, with just 20 patients plus 10 patients, Drug Controller General of India has approved the drug and is exempted phase 3. It is a dangerous sign, in the sense that even though it is there for already 6 years in the market, we do not know what else will happen, because there is a system evolved over a period. We want to bypass it today because we want to save the people. Assume that the drug is approved in a faster manner without doing a complete safety study. I want to tell two things. First thing is, having an efficacy we can do within 5 days. The person has got a disease, you give the medicine, if it works, within 5 days to 10 days, you know that it is working. But that drug does not cause any effect. It is actually years of study. For the drug, it could have helped you in the next 10 days to cure. But maybe it is damaged something which is visible after a long period, like cancer is induced now. You know only after 2 to 3 years. Something is inhibited, you may get diabetes after some time. Psychotropics, where it is so, even the safety study is done today, it is not complete, it is not comprehensive. They are able to test only the visible things and things as much as possible. If they start doing everything, then no drug will come to the market. Like no, teratogenicity. Next generation will affect or not. Mutagenicity. Whether it will affect children or not. We cannot take children as a volunteer and test it and prove it is dangerous or not dangerous. So it is a balanced approach as far as safety is concerned. So in my opinion, what is more and more dangerous is if you allow some drugs without doing a complete safety study, you may save the persons from disease, corona, or from infection. Sometimes without that also you may get cured because 97% of those who are infected, they don't know. And for those who are infected, maybe 97% are curing. So hardly 1 in thousands of people are only really succumb to the disease. But if the wrong drug is given, and he will get something which is not known, which will be known after some time, after that there is no cure. And if that happens in a worldwide, it's in a major population, it would have harmed the big population, much dangerous, more dangerous than corona itself. Like thalidomide, tens of thousands of children in Europe were born with malformed limbs and legs, and that is because what wrong drug was approved without complete safety study. So repurposed or original drugs. First, we should go for the drug which is proven for decades, not recently approved drugs. The drug which is approved in 2017 in Japan, that is being repurposed, which means it has not undergone phase 4 study. Phase 4 means what? In real world, you are launching and testing where it is dangerous. For the drug which is approved in 2018, there is no phase 4 study. There is no post-surveillance marketing study, which means its danger will come to know after use. So to escape from something which probably we can cure with simple treatments or even without treatment, we are taking something which could be actually toxin, which there is no proof that it is a toxin. We are thinking it's a drug. So that way, drugs are repurposed drugs. Unless they have crossed for a couple of decades, it is not advisable. Even if they crossed a couple of decades, see, people are getting diabetes. In fact, people are dying by ischemic heart disease. About how many years? Take about last 60, 70 years. Number of people are high today compared to 60 years before. That means, have we really improved? Answer is no. It is increasing means we don't know which drug which we are thinking safe is induced some other disease, and the disease we don't know source. Most of the diseases we are getting today, we don't know why it has come. It could be due to some other drug also. There is no proof today. Unless it is proven, we cannot doubt anything. But we have a disease, we go to the doctor. So most of the times we should rely on the natural medicine, natural treatment, natural food, uncontaminated things. So try to avoid the medicine as much as possible, even if safety is proven, because safety proving also has a limitation. Thank you.
Thank you very much, sir. It was interactive connecting with you. Thank you.
Well,
please. Yeah, please go ahead. Yeah. So actually, your area of problem is lungs today. That is an area of attack. Compartment of attack is lungs. So lungs should be as clear as possible. So just assume a highway. Highway is there, all vehicles are going and coming. Like in lungs, you have a highway where oxygen molecules are going inside and carbon dioxide molecule is coming outside. So highway should be clean and free. Assume that you are putting a debris on the sides of the road and making the road smaller. Naturally, your going and coming will be lesser and patient have a problem. And in the lungs, if your lungs airways are clean and healthy and active, even some inflammation occurs due to the entry of corona, that cannot block your way. And in few days, body will attend to it. And until then, you should be able to breathe properly. And those who are having this kind of other issues in the lungs, they also always have a risk of it. So keep your lungs clean and active. And you should, for that, what you should do? You should do pranayama, lungs exercise, or just you run it. When you run it, you have to breathe high and low, high and low, high and low. So in that process, body will ensure that your airways in the lung are kept completely clean. Why are you cleaning the home? So that we want to make the mobility be better. As far as your lungs airways are clean and wide and perfectly housekeeping is done, your chances of getting attacked by any organism will be reduced to highest extent. This is another way of protection. As I said in the beginning, hands, eyes and all. Second is keep your area of lungs perfect by doing the pranayama. If not, at least do running, jacking kind of thing where your lungs is working to its best. Yeah. Yeah. The 100%, even in September, India, it should not be announced because enough study will not be available. If it's announced in August 15, they are prematurely approving something which probably not good. I think. But second is vaccine is going to work or not only if we put in the real world we know, because there are already 7 or 8 mutations are there. Second, by the time we introduce this vaccine which is created from some type of virus, it may or may not work for the new things. So we don't know whether what will have a complete cure. Just give an example. I'm telling you, SARS virus which came in 2003, 2012 MERS came. SARS is what? Severe acute respiratory syndrome. Second one is Middle East respiratory syndrome. Both are coronavirus only. And there are many varieties starting from 1950s. Even today, there is no vaccine for SARS or MERS. How many years are gone? 16 years or 17 years have gone. Here for the MERS, it has gone 7 years or so. Further, even today, there is no vaccine. Vaccine could not be developed. Vaccine cannot be developed for those viruses which keeps on mutating. Here we can attempt, we can try, we can put it. Maybe at least today it may help us, but nobody can say that a vaccine can cure until it is put into the market. Yeah.
Yes, sir. Well, Mani sir, shall we wind up the session?
Yes, sir. Well, we can wind it up, sir. There are no more questions. There is some question they are asking about. It's not a clear one, I believe. It is something like aerosol. I don't know what they have meant. It says,
here,
please tell me the aerosol model is not tried injecting, mixing with oxygen for patients.
Yeah, actually here, I understand. So here aerosol means what they want to give is they want to go through the mouth or nose so that particles in the air, drugs in the air directly goes to the lung. So when you take a tablet or injection, you are going to the stomach and it comes to the blood plasma, or injection directly putting into blood plasma. And like small amount comes to the lung. Yes, sir, exactly, same thing only. Straight away goes to the lungs.
Lungs, correct.
Yeah, it is one of the steroids is being tried today for phase 3. It is there, I think phase 2, phase 3 to give us an aerosol as through a nebulizer, as you said, exactly, sir. That will be definitely a wonderful approach that is being tried.
Again, there is one, maybe the last question, sir, I'll read it out. Ishwar Chanda, sir, are clinical trials also conducted for repurposed drugs? I don't know why.
Yeah, of course. Of course, you have to do it.
Yes.
Because how do you know that coronavirus gets removed, COVID removed by the drug? They have to do clinical trial. Yeah, it is done. It's 100%.
Okay, sir.
Okay.
Yeah.
So we are.
Thank you. Thank you very much, sir.
End of the session right now.
Yeah.
Anyway, thank you, sir, for being patiently with us. I will just give I'll give this to PONNUSWAMY, sir. Thank you.
Yeah. Thank you. Thank you very much, sir. Yeah, thank you, sir. Thank you. You have some net issues, I hope. Thank you, Senthil. It is more than 2 hours. It's a very tiresome usual. Yes, you answered so many questions.
Excellent.
Really, really wonderful and useful session, really. And now I hand over the session to Professor Bindu Madam to propose the vote of thanks. Thanks for this.
Thank you, sir. It's my privilege for having been asked to propose the vote of thanks on this great occasion of the webinar. I, on behalf of PG and Research Department of Chemistry, Government Arts College, Coimbatore, extend a very hearty vote of thanks to our eminent speaker, Dr.U.P.SENTHIL KUMAR, Senior Vice President, Orchid Pharmaceutical Limited, Chennai, who has wholeheartedly accepted to be our guest for this virtual session, drug discovery and development, the challenges and opportunity to combat COVID-19. I must mention our deep sense of appreciation for giving an excellent coverage on these various drugs, its impact on living system, and the functioning of drugs. This is the need of the hour. It's interesting to note that many pharmaceutical companies of our country is actively participating in the field of research in drugs and its discovery, and Orchid Pharmaceutical Company being the one. Thank you, sir, for your an important, inspiring, illustrative, illuminative, interesting, and interacting presentation, which has given us the hope to combat any kind of pandemic now we are facing, as well as in near future. Cheating is a bad business, we all know, but we have found that by cheating only we can combat this virus, as you have told, sir. So let us make drugs which will cheat the virus and make this world COVID-19 free. Thank you very much, sir. My heartfelt thanks to all our participants in and around the state and over across the country, which includes ambitious and dynamic students, hardworking, challenging research scholars, and intellectual and creative faculty members. Thank you very much. Your participation is the key for the success of our webinar. Thank you very much. I take the privilege to mention my thanks to Dr. Mohan Raj, who is Assistant Professor, Manoன்மணி Sundarnath University, Constituent College, Marthandam, for readily helping us and giving us the technical support all throughout this session. My sincere thanks to Dr. Ratina Kumar, Assistant Professor of BPA, Manoன்மணி Sundarnath University, Constituent College, for supporting us by hosting this program through YouTube, which is accessible for all others who could not join us in this Google Meet. I must place on record my sincere thanks to our principal madam, Dr. K. Chitra, for giving us an opportunity and encouraging us to conduct the webinar during this pandemic session. This event has been the result of interest, commitment, excellent planning by our team leader and the captain of our ship, Dr. S. PONNUSWAMY, head of the department and associate professor of chemistry, for arranging this wonderful session. Thank you very much, sir. I thank Dr. Uma Rani, associate professor of chemistry, for giving a wonderful welcome address to everyone in this program. Dr. Velamani sir deserves an appreciation for beautifully handling the question answer session. Thank you, sir. Finally, from the bottom of my heart, I thank my faculty members and the friends of our department who are dynamic and enthusiastic, and they are readily available with the support and assistance, and they are the backbone of our department during these challenging activities. Last but not the least, we must all thank the science and technology and its development, which has brought us in this virtual session, otherwise which would not been feasible. I thank the Almighty for giving us the good health to acquire wealth of knowledge through such wonderful session given by our eminent speaker, Dr. U.P.S. sir. Thank you very much, sir. This session is a drop of water in the ocean of knowledge, and we expect more such programs should be conducted by our department and by our HOD, sir. Thank you, sir. Thank you all. Stay home, stay safe, and save the nation from this pandemic. Thank you, sir.
Thank you, Vidhu madam, for your wonderful vote of thanks. Thank you so much, and thank you all again as a convener. And one thing I wanted to mention, Dr. Mohan Raj is our alumnus. He did his MSc and PhD here. He is my MPhil and PhD scholar, and he came forward to support me for this webinar. So personally, I thank Dr. Mohan Raj and Dr. Ratna Kumar for their support, and again Dr. U.P.S. He is always here to support us. No doubt about that. Thank you all. Thank you all. I thank all the participants in this webinar to make it as a wonderful session. Thank you. Thank you all. Thank you.
Thank you, boys. Thank you. Thank you, boys. Thank you.